Next, I wanted to shift to a quick story about an email that I got maybe 2 or 3 years ago. It was from my friend Priyanka, who lives in Switzerland, and she swore that this family in London had a child with Kabuki syndrome, and I just didn't believe her because I've received so many emails across over the past few years in parenting my 6-year-old Walt, where people have told me that they know somebody else whose child might have the same condition as Walt, and it always turns out to be a different rare disease that starts with the letter K. But it turned out that my friend Priyanka was right, and I was able to meet Layla and Edo Zania, who have the incredibly adorable 5-year-old Roya, who also has Kabuki syndrome. And I was able to learn about Layla and Edo's incredible investments in something called the Roya Kabuki Initiative at Boston Children's Hospital. Edo's going to kick off after a short video to tell us more about this program. But the overarching idea here is this is an incredible example of how we're bringing innovation to this space, where we're able to have a clinical center of excellence where patients can go and see any specialist or many specialists who know Kabuki syndrome and deeply understand it, but then use that patient data and clinical observations to drive important research that will change the space. I have been so inspired to see this work with Dr. Bodamer and the Zania family, and I'm really excited for you all to hear about it today. So with that, let's kick off the video.
Big picture, when I look at a child, I see dreams, I see future, I see sincerity, I see laugh, I see none of the bad things that we see in the world. And so I want every child, and thinking about the next generation, everyone to have the opportunity to achieve all they want.
See the Roya Kabuki program as a program that has 3 main goals. One objective is to provide families with children with Kabuki syndrome with a multidisciplinary team approach. The second objective is to build a Kabuki community together with other parents, together with families. And then lastly is obviously to develop a therapy and further the research.
I know it's not easy to find a cure for some of the diseases that we know of. It makes me feel a strong sense of responsibility towards what I do, and it gives me great motivation to do something that I think will have an impact for someone. Talking to the people who are caregivers or are affected by a certain disease always makes me go back to the lab and say, I have to do this. And so it really does not matter how tired I am, or if I don't want to get out of bed, or if it's late in the lab and it's snowing outside, or whatever are the conditions, I always want to go back and do it until I get my result. And I really hope someday the ultimate goal is to make an impact in someone's life.
Our work is most focused on rare genetic disorders. There may be only a handful of children worldwide known with a particular condition, but if we think of these conditions as a group, they are actually fairly frequent. Obviously, we feel very passionate about Kabuki syndrome, but if we are able to identify a therapeutic approach that would address the underlying mechanism, that therapeutic approach could then be applicable to other conditions. So I think we might actually have a much broader impact than just on a rare condition like Kabuki syndrome.
I'm optimistic about Kabuki syndrome and other genetic diseases because I've seen over the past 20 years how much progress has been made in this field in general. And so I'm really optimistic that with more research, and not in a very long time, we will be able to manipulate things in a positive way so that some mistakes that are in the DNA in someone's cells can possibly be corrected. And if I can help, in whatever I do every day, that it would be definitely a great achievement for me. That's why I'm here.
Welcome to the Global Kabuki community. I can't stress enough how pleased I, with my wife Layla, are to see all these global efforts come together around a common goal. This is our story. I'm the proud father of Roya that you just saw in the video. She's a 5.5-year-old girl, and we live in London. My name is Eduardo, and my wife Leila, like many of you at the beginning of your journey, were given a diagnosis, a very scary one. Your daughter has Kabuki syndrome. After months of looking for answers, we were introduced to Boston Children's Hospital. And a German godfather, Olaf Bodamer, and our Italian godmother, Manuela Ghissoni, came to our rescue with open arms. You will get to know them in a second. We gathered our forces, we fundraised, and we convinced them both to dedicate their life to the study of this condition. And we created the Roya Kabuki initiative at Boston Children's Hospital. Fast forward 5 years, and here we are. Around 150 Kabuki kids are enrolled to date, and so many exciting therapeutic studies are on their way, and the team will be speaking to you shortly. And this is just the beginning, as we see. We started it as an interdisciplinary clinic for patients that have one point of reference to have your kids visited by so many different specialties, heart, endocrinology, immunology, and so forth. But soon after, we developed it into a therapeutic research center with one persisting mission: develop therapies for patients with Kabuki syndrome, and ultimately, clearly find a cure. But what struck us the most, and you can see it coming through very well in the video you just saw, is the fact that, and I'll use Emanuela as an example, this is not just working on a sample of blood in a laboratory. She's working for our kids. Before our visit to Boston, I've never seen a scientist come to a clinical meeting. I've never seen a lab so full of pictures of kids. The dedication of her and her team is mind-blowing, and dedicating one life for a cause for our kids. I just want to say one more immense thank you from all of us to all of you of what you do every day. I want to close my segment on how I started it, and in the same line that Annie was pointing out. We are today a global community. For exactly this purpose, Leila and I joined the board of the Kabuki Syndrome Foundation 2 years ago, because we wanted to ensure that programs like the Roya Kabuki initiative not only had the funding, but most importantly, had the coordination and collaboration across institutions worldwide. Because ultimately, and I think these couple of days showed together, we are a stronger group of people, of individuals. So without further ado, please let me present to you the dream team of the Roya Kabuki initiative, starting from its captain, Dr. Bodamer, Associate Chief Genetics of Boston Children's Hospital, Dr. Ghissoni, co-director of the program focused on muscle studies, Dr. Goodlett, neurodevelopmental champion for the Roya Kabuki program, and lastly, Tara Daly, program coordinator, who will speak about the opportunity for research participation. The stage is yours, and at the end, we will take questions for the group. Please. Thank you.
Thank you very much, Edo, for this kind introduction, and good morning, everyone. I'm deeply honored to present today on behalf of the Roya Kabuki team and to speak about strategies of drug development for Kabuki syndrome. I will never forget the moment when we first were brought into contact with Eduardo, Leila, and Roya, who inspired the Roya Kabuki program at Boston Children's Hospital. And it was really, in many ways, for me personally, a kind of a change in direction for clinical research and clinical operations as well. So the Roya Kabuki syndrome program or initiative was brought to fruition about 4 years ago. And it's really focused on patients and family. It takes a holistic approach. And one important aspect is really to understand what are the unmet needs of individuals with Kabuki syndrome and really rally around those unmet needs and focus on quality of life. The program is built on 3 pillars. First, it's focused on an integrated care model, bringing in other subspecialties that touch on Kabuki syndrome and building subspecialty expertise for Kabuki syndrome. And we have to date seen about 70 to 80 patients in this interdisciplinary clinic. And many colleagues of mine really commented on the fact that they have now built up significant expertise in Kabuki syndrome. The second pillar is to increase awareness for Kabuki syndrome and related rare diseases in the medical community and build relationships with local care teams and local hospitals to also share clinical care. And lastly, as we will hear from our team, we are also very much focused on basic and translational research with the ultimate goal to identify therapies and ultimately a cure, the moonshot for Kabuki syndrome. The team has grown substantially over the last years, and I just wanted to highlight several of the team members. You will hear in a moment Dr. Cusoni, good friend and co-director, who is a world-renowned muscle biologist. You heard yesterday from Max Hyman about his work creating a simple C. elegans model for Kabuki syndrome. We have also the support of Len Son, who is really a world expert on zebrafish, and we're working with him actively to create zebrafish models for different mutations in Kabuki syndrome. Xiaoting Ma leads the therapeutic development initiative in the program. Lucy Yongsok was a recent addition to the program. She is a very astute bioinformatician who helps us make sense out of the big data we generate. And in a moment, you will also hear from Benjamin Goodlett, our neuropsychologist focused on Kabuki syndrome. And lastly, from Tara Daly, the heart and soul, if you like, our program coordinator. Before I go into details, I wanted to kind of talk a little bit about the general concept of therapeutic approaches. Where can we really target therapies to? So there is obviously a mutant gene, so there is a genetic misspelling. That genetic misspelling may result in a change in the transcript. So genetic information is transcribed into what we call RNA. This RNA is then used as a template for the actual protein. So for a demethylase, for example, or methyltransferase, this protein obviously has an important function in regulating different pathways. So a mutation may also result in secondary pathway disturbance. And then obviously it results in clinical symptoms. So we can address therapeutic approaches at each level. You may have heard about gene therapy, gene editing, and obviously organ transplantation that targets the mutant gene or gene replacement. We are now also have tools available that would allow us to consider messenger RNA therapies where we replace the mutant messenger RNA. There are therapies for other conditions where the protein that's either dysfunctional can be replaced, so enzyme replacement therapy for other conditions. Chaperones that stabilize a mutant protein have been championed. We can also address the pathway disturbance, so kind of the downstream consequences of Kabuki syndrome using small molecule therapies. For other conditions, there are also dietary approaches and cofactor supplementation. And lastly, for clinical symptoms, obviously, as I mentioned, a multidisciplinary clinical approach. Now, what are some of the challenges for Kabuki syndrome? The Kabuki syndrome genes are large genes, some of the largest genes in the genome. So gene therapy at the moment with the currently available tools is unlikely to be feasible. However, there is an opportunity for gene editing, probably not for messenger RNA therapies, because again, the gene is extremely large and the resulting mRNA is also very large. Protein replacement therapies for the same reason, the resulting protein, again, being very sizable. does not make this approach amenable to Kabuki syndrome. So we're kind of left with a small molecule therapeutic approach, which I will show you in a moment how we are going about to identify small molecule therapies. Keeping in mind, this is a high level overview, so I will not go into too many details throughout the next couple of slides. So therapeutic development, it all starts with the patients, so gathering patient data, generating model organisms that reflect certain aspects of the condition are immensely important. And we have done this through collaboration with Max Hyman, as you heard yesterday, Alain Son with the zebrafish, and the mouse model that we established in the laboratory. And then there's also a unique opportunity to generate iPS pluripotent stem cells and have them develop into neurons for phenotyping. And this is an ongoing effort here at Children's. And then lastly, we are collecting clinical data on clinical phenotype, clinical specimens for a bio and data repository. And we look forward to the launch of the National Patient Registry and to the collaboration with All Stripes. The next step is really to think about what is the underlying disease mechanism and the different strategies that one can employ. And I'm just highlighting the ones that we focused on. We established a collaboration with Max's wife, Miriam at MIT, looking at neurometabolism in a animal model of Kabuki syndrome. And we are replicating that effort in human patients using a very sophisticated spectroscopy tool that is available at Boston Children's through a collaboration with one of my colleagues in neurology. Then Emanuela will present some data, promising data on muscle studies in a moment. We have, as I mentioned, Lucy working on analytics of big data. So these are data that are generated through the analysis of downstream changes that occur in individuals with Kabuki syndrome related to the formation of messenger RNA in downstream genes that are involved. And we also have a collaboration with an individual, Dr. Klisch at the Broad Institute, who actually looks at, who investigates and analyzes metabolic perturbations, so changes in metabolic pathways that might be affected through Kabuki syndrome. So these are ongoing efforts which have led to the identification of therapeutic targets and have also led to the to a high throughput screening effort to identify potential therapeutic candidates, and then obviously need to be validated in patient cells and or of course model organisms. And this is done through a mechanism, through a tool called high throughput small molecule screening. And let me take you through that with this slide. So high throughput screening is a method to identify small molecule therapies using a system where cells are exposed to drug candidates, and these drug candidates can be either FDA approved, existing FDA approved drugs, can be vitamins, for example, or naturally occurring compounds, and can also be exposed to chemical compound libraries where only the chemical structure of the potential drug is known. The way we have set this up is we have generated two unique cell lines, one for KMT2D and one for KDM6A, that are tagged with a fluorescent reporter. The fluorescent reporter can either yield a fluorescent red color, as in this case, or can yield a fluorescent green color. This, at the moment, unfortunately, I'm not able to share any details because we are currently in the process of submitting a patent, but hopefully we can make this available more broadly. The idea behind this screening is you see these little circles on this plate. These little circles contain these cell lines that we have generated, about 1,000 to 1,500 cells with these unique features in each well. And these wells are then exposed to different drugs. So for example, you see there's a drug set 1 to 16 here in different drug concentrations. When there is a drug that changes that unique property, that changes in simple terms, the Kabuki KDM6A related phenotype back to a normal state, you will see an increase, in this case, in red fluorescence. So in this case, the circle here with the red color, that's a drug that's potentially a candidate for reversing the phenotype in Kabuki syndrome at the cellular level. And then you see other wells that are yellow. So this would be an intermediate response. And then you see the ones that are kind of blue or green, which there is no response identified. In any case, we would then take those therapeutic candidates, these responders, and repeat the entire screen with a more closer look to the actual drug concentration. And if it's replicated, then the next step would be to take this drug candidate to a validation system. This could be either a cellular system or an animal model before additional preclinical studies take place. I think we all have to appreciate this is a high risk screening with no estimate on how high the probability would be that we will identify a therapeutic candidate that will eventually make it to a clinical trial. But based on the results we have seen so far, we are cautiously optimistic that this might actually pan out within the next years or so. With this, on the therapeutic development side, one has to, in parallel, and there was already some discussion around that, one has to gear up for clinical trial readiness. So one has to really identify and characterize the natural history of Kabuki syndrome. One has to identify the clinical outcome measures. What are the outcome measures for a clinical trial that on the one end meet the needs of the patient community, but are also outcome measures that are amenable to drug treatment. And this all together obviously is a kind of are two important aspects that are supported by patient advocacy like the Kabuki Syndrome Foundation and ATK. So input from patients and families is critical for that aspect. And then obviously, this all needs to be done in partnership. This could not be done at a single center. This has always the kind of a multi-center cooperation with pharmaceutical industry approach, which then would hopefully lead to a clinical trial soon. Let me kind of summarize what we are doing. We are kind of using an integrated approach to identify therapeutic targets and develop therapies. So when I say integrated approach, this goes across multiple layers of information and data, starting with genomics data, so changes in genetic information, epigenomic regulation, transcriptomics. So these are the changes in downstream gene expression, changes in the proteome, changes in the metabolome. And then obviously we are also looking at different phenotypic aspects across the cells, the animal models, and the patients. So this is an ongoing effort, and I don't want to go or spend too much time on the details across the entire program with different disease models and ultimately therapeutic development. So with that, I'd like to close, but I also want to acknowledge that obviously I'm presenting on behalf of a fabulous team here, and I'm most grateful to my team members, to Emanuela Guzzoni and Max for his work on C. elegans, my colleagues across Boston Children's and across the network that we are involved with. And of course, also I'm most grateful to be given this opportunity to today through the Kabuki Syndrome Foundation. And I'm also grateful to ATK. Before I close, let me just touch on my inspiration or our inspiration. And it all started obviously with Roya. I'm glad, Eduardo, by the way, that I'm the godfather. I didn't realize that, but I'm more than happy to take on that role. And I'm also most grateful to be part of this community. It feels like being part of a large family. And again, I'm most grateful for that. With that, I'd like to close and hand over to my good friend, Emanuela Cusoni, who will speak about muscle studies in Kabuki syndrome. Thank you.